Good morning students. Our today's topic is bioremediation. Bioremediation, nothing but remediating something with the help of biomaterials or bio things. That is, remediation is to solve a problem. Here we are going to solve our environmental issues with the help of microorganisms. The definition of bioremediation, it refers to the process of using living microorganism to remove the environmental pollutants or prevent the pollution. Environmental pollutants such as heavy metals, organic waste, pesticides, that kind of pollutants will be remediated with the help of microorganism. The removal of organic waste by microorganism which makes the essence of bioremediation in the environment. And the bioremediation technology by using microbes was first invented by George M. Robinson. Next we are going to see about types of bioremediation. The two major types of bioremediation are, first one, in situ bioremediation. The name itself implies in situ in the sense in the site itself where the pollutant occurs. In situ bioremediation means there is no need to excavate or remove soil or waste in order to accomplish remediation. There is no need of removing the pollutant from the environment. We can remediate the problem in the site itself, that is in situ bioremediation. It employs use of harmless microbial organisms to degrade the chemicals such as pesticides or heavy metals or organic waste. And sometimes it involves the supplying of oxygen and nutrition to enhance the activity of indigenous microbes in the environment to remediate the pollutants. And also it includes the circulation of aqueous solutions through naturally occurring microbes to degrade organic compounds. And this is the picture, it shows the in situ bioremediation process in the site itself. Types of in situ bioremediation. There are two types of in situ bioremediation. The first one is intrinsic and then second one is engineered, and it is also called as enhanced in situ bioremediation. The first one, intrinsic in situ bioremediation, it is nothing but conversion of environmental pollutants into the harmless forms through the innate capabilities of naturally occurring microbial population. It is nothing but we are not going to give such oxygen supply or nutrient supply to the indigenous microorganisms. Naturally the activity of microbial population will degrade the organic compounds. There is no external source given to the microorganisms. And then engineered or enhanced, here the microbial activity will be enhanced by some external parameters. Here it accelerates the degradation process by enhancing the physico-chemical conditions, nothing but the physical conditions like water condition, moisture condition, temperature condition, and the chemical like nutrient supply will be given to those groups population, and it will encourage the growth of microorganism and also the potential of microorganisms in degrading the environmental pollutants. Next one is advantages of in situ bioremediation. Here it does not require any excavation of contaminated soil or water, and it is very less expensive, less chance of contaminating other sites. Because this bioremediation will be carried out in the site itself, there is no need of removing the contaminated samples, that may be soil or water, from the site. So we can carry out the bioremediation process in the site itself, and it is also very less expensive, less chance of contaminating other sites, and it is easy to treat a large volume of soil at once. And coming to disadvantage, it is a slow process because huge volume of the sample will be processed. That is why it is a slow process and difficult to manage, and it also requires uncompacted soil because it requires saturated soil. The next type of bioremediation is ex situ bioremediation. It involves the removal of waste material and collection to the place to facilitate microbial degradation. Here the sample should be removed from the site and it should be transported to some other site for the processing. And the average time frame of ex situ bioremediation is 60 to 90 days. This is the most advantage when compared with in situ method, because the time taken for the bioremediation will be 60 to 90 days, because in situ bioremediation a large volume of sample will be processed. So, it takes a longer time. Here it takes a much lesser time when compared with the in situ bioremediation, and better control area, depth, temperature, nutrients, oxygen, moisture, etc. Here the external parameters like temperature, nutrients, oxygen, moisture can be easily controlled for the process. On the basis of contaminated area, it is also divided into two types. First one is solid phase system. Under solid phase system there are three processes. First one land forming, second one composting, and third one biopiles. In land forming, the contaminated soil will dig up and it will be spread over as a bed, and it will be tilted until the degradation process will be carried out. It is the process in which the land forming is taking place, and then tilting of soil is carried out to remediate the contaminant in the soil. Then second one, composting. Composting is nothing but conversion of waste material into useful material. Here the organic waste will, the agricultural waste, it is subjected to composting. Then biopiles. In biopiles we can remediate the petroleum hydrocarbon pollutant. Here we should supply oxygen and nutrients to the microorganisms. The next one is, and this is the picture's representation of biofile. Here these hydrocarbons will be remediated by using microbes with the help of oxygen supply and nutrient to the organisms. The next method is slurry phase system. It is more rapid process when compared with of solid phase system. Here the contaminated soil is combined with water and other additives like nutrients it will be carried out in the bioreactor, and then maintenance of optimum conditions will be carried out. It includes bioreactor, bioventing, biosparging and bioaugmentation. The first three bioreactor, bioventing and biosparging, it comes under biostimulation, that is stimulating the microorganisms to degrade the pollutants, and bioaugmentation, this is nothing but the supply of some exogenous microorganisms to carry out the bioremediation. First one, bioreactor. Here the waste processed through an engineered containment system, and it will be carried out under controllable condition, and it can be predictable. Then second one, bioventing. Here it employs the use of air flows. The airflow rates will be very low, and oxygen and nutrient supply will be given to microorganism to stimulate their activity. This is the picturized representation of a bioreactor. This one is biosparging. Here injection of air under pressure will be carried out. Here underwater table, the air pressure will be given to the underwater table to degrade the hydrocarbon pollutants. And then bioaugmentation, before that the degradation will be carried out only by the indigenous microorganism. In bioaugmentation we will be adding some exogenous microorganism, that is adding microbial culture to enhance decontamination. This is a picturized representation of biosparging. Here the groundwater oxygen level will be increased by giving the oxygen supply. So, biological degradation of contaminants will be taken place. Here there are some microorganisms, some examples of microorganisms that will be involved in bioremediation. For example, aerobic microorganism, alkaligens, Sigmomonas, Rhodococcus, Mycobacterium, Pseudomonas, etc. are involving in bioremediation. And example for anaerobic, Delohalococoids, and for example lignolytic, Phanerochite, Chrysosporium. Factors affecting bioremediation. nutrient availability, moisture content, pH, temperature. These are all some factors which affects the bioremediation process. If the nutrient availability for the microorganism is high, the process takes place by the microorganism will also high. For example, for petroleum hydrocarbons, we need to supply nitrogen source to enhance their potential, and then moisture content also given to the microorganisms to carry out degradation, and pH, the pH range should be between 7 to 8 in case of bioremediation, and then temperature. Coming to temperature, most of the bioremediation process will be taking place by mesophils, that optimum temperature range is 25 to 45 degrees Celsius. And next one is phytoremediation. Phyto, the name shows plant based. Here the soil, water, heavy metals, petroleum hydrocarbons and inorganic pollutants are degraded by the plants. The plants involved in the phytoremediation is sunflower, Indian mustard, etc., and it is a less expensive bioremediation technique than any other type, and also it is based on an in situ technique and does not require transportation or other complicated treatment procedure. It is phytoremediation is an in situ remediation process only, and then the toxin become a part of food chain. It removes contaminants from the surface only, and a bit slow process, only surface contaminant will be removed by the phytoremediation process. And then mycoremediation, it is a fungi based remediation. As like bacteria, fungus also involved in remediation process. Here water bodies, soil, crude oil, industrial effluents such as PCBs, heavy metals, dioxins, etc. can be treated. It is an eco-friendly technique and it is also a cheap and cost effective method, but the process is slow. This is the only disadvantage of this method. Then microbial bioremediation is nothing but bacterial based, as we have already seen. Here the microbes involved are AcidothioBacillus ferrooxidans, Pseudomonas putida, etc. Here it involves both in situ and ex situ techniques. We see that phytoremediation involves only in situ technique. Here the microbial bioremediation involves both in situ and ex situ technique. Microbial actions in this type of bioremediation require certain conditions, that is the parameters given by us. Then it requires field monitoring. Advantages of bioremediation: completely natural process with almost no harmful side effect. Here because only we use the microorganisms which are harmless to our environment, and it can be carried out in situ for most application with no dangerous transports, and then it is a quick turnaround time to make soil and water useful. Minimal equipment needed except for specialized species like bioreactor, bioventing and biosponging. That is all about the advantages of bioremediation. So, today we saw about bioremediation, its definition, types and what are the methods used in bioremediation. Thank you.